This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 774, recorded on June 29th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 87 Fahrenheit, and they say that feels like 99 Fahrenheit, and it's supposed to storm in the next little bit, so you might lose me. I don't know. And that's the same as 34 Celsius and feels like 37 Celsius, so pretty hot.
Yeah, it's pretty hot here in New York City. 35 Celsius, we have a heat advisory. Let's see what it is in the rest of the country. Coming back from Tulane University, Bob Garry.
Vincent, good to be here again. It's 29 degrees C here in New Orleans, and it's a little bit of precipitation coming down. Not too bad, though.
Not bad. All right. And joining us for the first time from Scripps Research, Kristian Andersen. Welcome to TWiV.
Thank you, Vincent. Thanks for having me on. Yeah, and being in San Diego, 20 degrees C, 70 F. But it's actually a little cloudy right now, which I'm pretty excited about. I suspect it'll be sunny within the hour.
Nice.
Yeah, you're in the season of night and morning low clouds, right?
That's right. It's the June gloom.
Mm-hmm.
All right, we have a mission today, and that's why Bob and Kristian are here. We're going to talk about a manuscript that made a lot of noise. I don't know, last week, I think, or the week before when it was released by Jesse Bloom. It's called Recovery of Deleted Deep Sequencing Data Sheds More Light on the Early Wuhan SARS-CoV-2 Epidemic, preprint at bioRxiv. And Bob and Kristian have agreed to come on and help us unpack this. But before we do that, Kristian, your first time on TWiV, why don't you give us a little bit of your history, where you're from, where you got trained, and so forth?
Sure. Yeah. So I'm from Denmark, and I did my undergraduate degree in Denmark in molecular biology in Aarhus. So not Copenhagen, the second biggest city in Denmark. And then moved on to do my PhD in immunology at Cambridge University and the MRC Laboratory of Molecular Biology, doing a lot of mouse work and understanding immunity with a focus on T cells. Sort of got a little jaded with that and wanted to do something a little more exciting. So I hopped over to the US, Boston, did my postdoc at Harvard with Padis Sabeti, also where I met Bob Garry, of course. And I've been working on infectious diseases with a strong focus on evolution, genomic epidemiology, understanding transmission and spread and evolution of these viruses. And now I'm a professor here at Scripps Research. I've been for about 6 years now, I think. And our focus has been on viruses as well. And again, a lot of this work together with Bob.
Yeah, as I told you yesterday, I visited Aarhus. Man, they taught me how to pronounce it.
It's hard.
I was getting it all wrong. And I think October 2019, and I guess you knew some of the people I had on. I did a TWiV there. It was really fun.
Yeah, it's a great place, and I still keep the connection back to Aarhus. I have some good colleagues and friends who are there for sure.
So I remember I flew in. I flew to Copenhagen, and then I took a train, and then my host was a graduate student, and she had heard I was going to Copenhagen for a meeting, I think European Society of Clinical Virology. And so she said, come to Aarhus for a seminar. So I did. But on the plane, I was reading in the magazine this picturesque street in Aarhus with all these pretty houses. So I took a picture of it on my phone and I gave it to her when I arrived. I said, I want to see this. So the next morning she took me there. It was this tiny street of little houses. You must know it, right? It's beautiful.
Yeah, it's a lot of character in parts of that city for sure.
Great.
And it's near the forests and the ocean. So yeah, forests and trees is something I'm missing here in San Diego for sure. Need more of those.
So I went to dinner with some students and they all said, this is the best place in the world to live. including, I think one of them was from Israel. He said, this is the best place in the world. And my host said, except it gets dark pretty early in the winter. You have to deal with that, right?
Well, if you like sunshine, I'll say San Diego is probably better than Aarhus and Denmark. But yeah, it's a good place, and science is really a big thing in Aarhus. And I'll say I go home every once in a while these days, and the place has exploded. It's much bigger now than when I left, which is many years ago by now. It's almost 15 years ago.
I think there's a Nobel laureate out of Aarhus, right?
Yeah, there are a few. Of course, you ask me now, and I'm blanking on the name, as I shouldn't. But yes, there's been a long history of especially membrane proteins. Actually, is one of the big things. First from physiological aspect, which is what the Nobel Prize was in, and then more recently around structures and understanding their function in both neurology, but also more recently in infectious diseases, of course.
Now, I noticed when COVID hit that Aarhus was mentioned often as helping, screening, sequencing, whatever, which was quite interesting, right?
Yeah, and Denmark in general, I'll say in terms of rapid screening, Denmark was actually screening almost 10% of its population daily, which I think is one of the main reasons why they were quite successful at keeping numbers low despite being pretty much fully open. And also importantly, that genomic surveillance program, they're sequencing most cases these days, which really give us a very sort of clear look of evolution, variants, and all these things. So very, very impressive. And it's small operations. It's a single university. It's a single lab doing most of the genomic sequencing for the whole of Denmark, which is really impressive.
Yeah, I met someone at another meeting, actually, from the State Serum Institute, right? And I guess it's in Copenhagen, right? And he got involved with the Mink outbreak.
Yes.
I can't remember his name. You probably know the guy.
Maybe Anders Fomsgaard, maybe?
Fomsgaard, yes, exactly.
Yeah.
And he gave me his book. He wrote a book called "It's Just a Virus," and it's in Danish, of course, but it's got a funny picture of him on the cover in some kind of, high containment suit, but he has regular shoes on. Picture.
Yeah, there's an article with him that I saw just a couple of days ago where he's sitting in his full suit, but then on the phone in his office. I'm like, that doesn't really make sense. But I guess it was a good photo op. Yeah.
Well, I tell you, when the Mink outbreak happened, I emailed him, and he immediately answered, and he gave data. He said, we're going to publish daily updates. It was very forthcoming. It was really good.
Yeah. And I'll say, actually understanding emergence and origins of SARS-CoV-2, I would say the whole Mink stories that we have seen both in Denmark but also in the Netherlands, where clearly this virus is very generalist, that's capable of jumping right into a mink population, burning through that population, right back out to people again, with very little evolution at all. There's a single mutation that we see pop up frequently, but it's fully capable of doing that, which is. There's other viruses that can do that, but I would say SARS-CoV-2 certainly has a unique ability to do that with a lot of different animals, mink included.
What's the story with the mink in Denmark at the moment? Did they quell the outbreaks? Are they still having the farms, or what?
They culled the entire population.
Wow.
I think there might be some small-scale operations, but... it was chaotic, right? And it wasn't done optimally, no question about it. But having this sort of parallel epidemic going on in animals is, of course, a risk, both in terms of viral evolution, but also spill back into humans from the mink population. And I will say one of the things that I was certainly concerned about too is going from mink to other farmed animals. Mink farms are often together with other farmed animals, and you can imagine SARS-CoV-2 getting established not just in the mink population, but also in other farmed animals, and maybe even rodents and other animals, which will, of course, would be a big problem in terms of keeping numbers low, eradication, vaccine strategy.
Are there wild mink in Denmark?
There, I believe there's some wild mink. There's also escaped mink, but of course Denmark is not. Denmark was the largest producer of mink prior to the pandemic, Of course, there are many mink farms here in the US, for example, too, is that there's...
And in China as well, yeah.
And in China, yes, absolutely. Yeah.
I'm curious to see how many wild animals end up being infected. I assume people are doing surveillance. I know Tony Schounts out in Colorado is interested in the deer mouse population in the wild, right? Yep. But I wonder what else, right?
Yeah, I mean, there are papers showing that the variants of concern, probably because of that N501Y mutation and possibly others, but are capable of infecting rodents, right? Which the sort of vanilla Q1 SARS-CoV-2 was not capable of. So, I mean, the host range is clearly broad. The question is if it's capable of establishing itself in some of these animal populations is certainly concerning. And I would say also just spill back into bats. Is that even possible? I'm sure Tony would have some good insights on this. Probably not super high risk, but certainly as the pandemic continues to be ongoing is one of the things that we need to be aware of.
Yeah. No, I'm sure we'll find out in the coming years. Absolutely. All right. So before SARS-CoV-2, Kristian, what were you working on mainly? What viruses?
So, I mean, most of our work is focused on Lassa fever and Lassa virus, and that's a lot of the work with Bob. Of course, Bob has worked with, our collaborators and colleagues in Sierra Leone for many years, and also colleagues in Nigeria and other places in West Africa. So Lassa has been and continues to be a strong focus of both of us and our collaborative projects, which are huge. Ebola, of course, is something that we were right in the middle of it when that started it's detected back in West Africa in March of 2014. So a strong focus on Ebola back then, and this continues to be a focus today. Zika, as that hit the Americas, and our focus was mostly on how did it get to the US, how was it spreading in the US, how did it emerge? And then more recently, also West Nile has been a focus of ours, which is an endemic virus here in the United States. We're working with a lot of public health partners, vector control labs all across the country here. Also working with academic institutions to understand not these outbreak viruses, because much of our work has been on outbreak viruses, but on getting better assessments of evolution, spread, transmission, what are the drivers on an endemic virus. That's the recent endemic virus, right? As you well know, came into New York City and was first detected there in 1999 and has since spread all across the country. So that's a strong focus.
We have to get you back when Dixon Despommier is here and talk about West Nile. That would be fun, because he wrote a little book about West Nile called West Nile Story, I think it was. And he loves to tell stories. The Ebola leads me to ask you. So there's a current outbreak, which I think the virus is very similar to the last one of 5 years ago. It seems to have persisted in someone. What do we know about that?
Yeah, that's the recent reemergence, and Bob knows this well too. In Guinea. Which is a continuation of the outbreak there, the epidemic in West Africa, which the last cases were described in 2016. What they managed to do and this is work done in West Africa, sequenced the virus. And what they realized is that it had a very low molecular clock. It connected all back to that Macona variant, which caused the epidemic in West Africa. And really importantly, had this one single mutation, the A82V mutation, which is something that developed during the West African epidemic. The new virus here has that signature mutation, as well as on a phylogenetic tree can clearly connect it back to so that is almost certainly a persistent infection, which of course we have seen in the DRC more frequently these days too.
Yeah, there was another outbreak this year in the DRC, right?
It's the same thing.
We had very strong epidemiology there on that one because, the first case, her husband was a Ebola survivor, and they could clearly link the two viruses there and show that reduced molecular clock.
Yeah.
So it's happening. People are sort of like a reservoir, right?
Yeah, I mean, I think it's interesting because, to me, it's humbling because if you had asked me in 2014, do you think Ebola can stick around in a person for 5 years and then cause a new outbreak? And I would have said, absolutely not. But it's clearly happening. And we saw several versions of this during both the West African epidemic, but also later in the DRC. They weren't quite 5 years, but they were long periods of time. And what's interesting here is that the virus during this period of time, it really doesn't evolve very much, which is interesting because Ebola has a very clock-like structure. We can see its evolution during an epidemic. It's very predictable. Even between epidemics, it's highly predictable. But when it finds itself in these latent infections or whatever it is in survivors, is that it sticks around and it doesn't really change much, as in it doesn't evolve at all. And that's a clear signature, which is interesting because it's the opposite of what we see with SARS-CoV-2, where we think that there might be these persistent infections that actually have accelerated evolution. I would say the emergence of the Alpha variant, the B117, first detected in the UK, is an example of that, where the molecular clock wasn't lower, it was much higher. So there's clearly differences there, which we don't fully understand, but is important and interesting for sure.
In the case of Ebola, do you think it's something where the virus is very slowly replicating and or truly latent? Or, is there any evidence that you can get?
There's definitely clinical latency, right? Because the patients are not showing virus, at least in the periphery. But, are there sanctuaries where the virus could be replicating at a lower level, say in men in the testes, for example, or in the CNS where, it's possible it could maybe come back and forth. One of the important things, though, is it does seem to be linked to immunity. So it takes a little time and for the viruses to reemerge from a person that is clinically latent. And so there's a thought that perhaps, that vaccines could be used to maybe control that in survivors. And I think it's important for the audience to know that this Ebola survivors in Africa, we need to be paying a lot more attention to those people. They have ongoing medical problems. This just reminds us that those survivors are important if they particularly can be a source of a new outbreak. So we need to really be, working with the countries there that have large numbers of survivors, maybe get them some vaccines that might be able to suppress the virus and keep it from being passed on to another person. Those are questions that, require a lot of research, but, they're definitely important to be doing those kind of things.
Great. All right. So you see, folks, virology is cool. A lot of things we can talk about. All right. So back to this preprint. Posted on June 22nd. Well, I guess that's the one I have anyway. I guess that's when I first saw it. June 22nd, it's probably been updated. And as I said, recovery of deleted deep sequencing data sheds more light on the early Wuhan SARS-CoV-2 epidemic. So let's start so this is about some sequences that were supposedly deleted from what's called a sequence read archive. So maybe we could start with explaining what that is. I don't know who would be better. Bob, or
let Kristian do that. Yeah, for sure.
Yeah, the sequence read archive is under the NCBI, which is a U.S. resource, U.S. control, which is the ability to submit raw sequence data. So sequences come off a sequencer, and then you take that data, that's the raw data, you generate genomes based on that. Or in this particular case, amplicons, because it was actually for the purpose of developing a new diagnostic that was using the nanopore platform. But the raw data can then be submitted to what's called the SRA, an archive, which is something that SARS-CoV-2, for example, many labs do, but many labs don't. I will say probably most labs don't actually submit to the SRA because it's the raw data. It's messy, it's big, it's difficult to submit, it's difficult to manage. Most people focus on submitting the actual genomes, mostly full genomes, and then do that to GISAID, which is a different different entity, as well as some time to NCBI GenBank too. So the SRA is just a place to put the raw sequence data.
I would say of the almost 2 million sequences in GSAID that, I mean, the vast majority of them do not have those raw data, the sequence read archives posted or any on NIH or any other database like that. It's actually kind of a rare thing to do. I mean, it's not a bad thing to do. I mean, you should probably be encouraged to do it. It helps, but it's not the usual thing to do.
Yeah, I'll say, we definitely all should submit the raw data. And I would say from my own group, we do that, although we are much delayed pushing it to the SRA. We do it on Google Cloud, for example, and make it available in real time there. But to Bob's point, like probably 95% of genomes that are there probably don't have the raw data on the SRA. But it is absolutely, and I mean, I totally agree with Bloom on this, that it would be advisable to have all the raw data available on things like the SRA. And I think it's important so you can test and check how good the genomes are, what confidence do you have specific genomes, are there concerns about contaminants and sequencing errors, for example. Absolutely. But it needs to be clear that for most cases, it's not really something that's being posted for most projects.
So this would be all the sequence. So any contaminants in the sample would also be uploaded as well, right?
But possibly, yeah. I mean, it depends on exactly what the source is. For many people, for example, you assemble your genome, so you align reads to your genome, and then you submit all the reads that aligned, which is then only going to be SARS-CoV-2 reads.
Sure.
But in other cases, metagenomic sequencing, where you sequence the virus, but you also sequence whatever else is in there, which is then submitted. So it really varies depending on what the specific project.
It's a highly specialized database, though. I mean, unless you're doing genomics and sequencing and actually looking at these questions about things like the SNPs and how deep are the reads and things like that. I mean, the vast majority of even virologists or people even studying the viruses wouldn't go to the SRAs to look at that original data unless there was just a question that came up about it.
Yeah. Okay. Got it. All right. So what sequences are Jesse saying were removed? Who put them there, and what was the purpose, and so forth?
Yeah. So I mean that's the authors of the Wang et al. Authors that had a preprint back in March of 2020. Mm-hmm. The paper is about developing a nanopore diagnostic. They amplify up some amplicons, some part of the SARS-CoV-2 genome. And then based on that, they can see, well, if you get an amplicon, that's a positive sample, but you can also get some information about specific mutations that might be in the genome. Those genomes are woefully incomplete. It's only, about 1 kb or so, and the full genome is 30 kb, right? So it's a really small part of the genome, but includes part of the spike gene, for example, in which we see a lot of SNPs. So that's what the paper was, and the paper is about the diagnostic. They have a table 1 where they actually show all the SNPs that they find in the data. That's both in the preprint and in the later paper, which was published in a journal called Small, which I believe is mostly a chemistry journal. It's not a no-name journal, as some people have assumed. It has an impact factor above 10, so it's actually a pretty high impact journal, and it's special for these kinds of projects, right? Where you're trying to develop a new diagnostic.
Yeah, I actually looked that up, Kristian, and there's been an insinuation that publishing in this journal Small, which is it's nanopore sequencing, so I mean that's a good place to publish something about a diagnostic using nanopore sequencing, right? But it actually has an impact factor of 11.5. Now, if you look at the top 3 general virology journals, Journal of Virology, Journal of General Virology, and Virology, that the impact factors of those journals do not add up to 11.5. Okay. So, I mean, if the insinuation is that the authors were hiding this data somewhere, I mean, the place to hide it is not in a journal that has an impact factor that's, higher than PNAS and a few of other very important journals. It's small is pretty well read. So they're diagnosticians. They're developing diagnostic assays. This is a perfectly reasonable place for them to put that data, and they weren't trying to hide anything.
So these authors uploaded the data for their paper to the SRA, correct? Which is something they decided to do. And then, according to Jesse, some of it was taken down. Is that right?
Yeah. So, and then they later deleted that. So I believe, and we actually have, because Bloom just updated the preprint, and we actually have the email from the authors to the NIH, which they have shared with Jesse. So we know what reason they gave, and the reason is that, I'm just reading from that email now. It said that I found that it's hard to visit my submitted SRA data, and it would also be very difficult for me to update the data, which is all correct. I have submitted an updated version of this SRA data to another website, so I want to withdraw the old one on NCBI in order to avoid the data version issue. So, and this is in June, so I believe around the time where they published the final paper in small. And I will say, deleting data is obviously not optimal. That's not what we should do, right? Data, we should put it online. We should make it open access. We should have it all there. But this is not unusual. This happens often. And the data version issue is a real one because the SRA is difficult to access. It is difficult to update your data on there. So if they wanted to put it on another website, again, not ideal, right? It should be available, but that's a perfectly legit reason for why you might, and certainly the data version issue is a perfectly legit reason for why you might request that it get deleted by NCBI, which is, when I read this, is like, yeah, I mean, that makes sense.
Would there be a way to? I was going to ask if there would be a way to cross-reference it to leave it on the SRA, just in general going forward or whatever, leave it on the SRA, but then have something that says, for more manageable or different or something, see XYZ location.
No, you would probably delete it under those and then maybe create a new bioproject if you wanted it on the SRA. Of course, again, these are from Chinese authors. I should say that they say they wanted to put it on another website. Maybe they have done that, but we don't know what that website is. We don't know. It might not be a public website. Things like that we don't know. And again, it's not optimal, right? But it's not suggestive to me that they're trying to hide anything, because again, this is commonplace. This is something that happens often, not just for SARS-CoV-2, but for sequencing projects in general.
Yeah. And I think the other important point is that in table 1 of that Wang paper, all the important data is there. Right. I mean, the SNPs are there. You can totally reconstruct everything about those genomes with the data that's in table 1. And in point of fact, you could do all the analysis that Jesse Bloom did in his preprint using that data from table 1. So,
I mean, in fact,
it's not what they were trying to do.
Table 1 in the Wang et al. Both preprint and paper is basically table 1 in Jesse Bloom's own preprint, right? Which is reflecting the same SNP data.
Right.
I agree that it's not optimal, right? Trying to pull the data from a table to reconstruct genomes is not optimal. It's better to have the genomes, but neither is it optimal to go back to the raw data and try and do it from there, right? None of these things are optimal. And yeah, small is not the optimal journal for this. But again, this is, science is rarely perfect. And there's nothing unusual about this. We can all agree that, yeah, there will be better ways of doing that. But that doesn't mean that because people didn't do it in the way in which we think would be the optimal way, that doesn't necessarily mean that they have something to hide and trying to obscure anything, which I think is a problem in this particular case.
And I mean, I think for these authors, the journal small is a perfectly reasonable place to publish it. I mean, it's got a really great impact factor, and they're probably targeting the audience that they really want to target with that journal. So, yeah, nothing surreptitious there.
I just want to point out for our listeners that SNPs is a shorthand for single nucleotide polymorphisms. So if the SNPs are listed in a table, then all of the sequence around those SNPs is wild type or consensus or something like that. And so that's how you would be able to construct what the rest of the sequence is. You know what the single nucleotide polymorphism or change is, and from that, then you could generate the rest of the sequence.
Yeah. So another, and Kristian just mentioned, and I haven't gotten a chance to look at the revisions that Dr. Bloom made in this preprint yet, but I did notice just the first look at it. One thing that he did do was to delete figure 2 of his first version, which was an email that was obtained by a Freedom of Information Act request from, I think they're an animal rights group. I'm not sure exactly what their role in the origins are, but they had obtained an email of another group who had submitted some sequences about pangolin coronaviruses, and they had asked for part of that data to be removed from the archives. I think that it was not. Kristian may know better than me, but it's still up there on the NCBI sequence archives.
That data had actually turned, that they were concerned about contaminants in that. So they didn't, which that happens often, I should say, like these big sequencing projects where you're sequencing a lot of viruses. Contaminants is a common issue you have to deal with, and you might not immediately realize that it's contaminated. I think a good example of this is, we used to sequence a lot of, when I was at the Broad, we sequenced a lot of Lassa and Ebola, they still do. And those reads ended up in some of the GTEC data sets, which is completely unrelated, right? And all of a sudden you realize that these people from Boston that have Lassa, only they don't, they're contaminants, right? So in these cases, there's saying like, look, we need to remove that so it doesn't confuse people and re-upload the data and we'll do whatever. And that I believe is what happened with these pangolin coronavirus papers, for example, which of course is totally unrelated to this particular study here.
Yeah, so I'm glad to see that that Dr. Bloom deleted that figure 2, because I felt that was one of the things that was really totally inappropriate with that preprint in some ways. So first of all, the authors were Chinese, so you're using an example of a removal of a sequence arguably related very closely to the origins of SARS-CoV-2, and the only reason that example existed was because of this Freedom of Information Act request, clearly political in nature, and to include that in what's ostensibly a scientific paper, I think that those are the types of things that scientists need to avoid. I mean, science and politics don't really mix, and we want to try to look at the data, that's available, but to put something in that just points to Chinese as covering up and doing all these things, which it's. The Chinese government has not been totally open about the pandemic. There's a lot of things out there that we would like to know and that hasn't been described. But to really hang it on some scientists that didn't have anything to do with this paper that he's making a case they pulled the data and that kind of thing. I thought that was one thing that was totally inappropriate.
Was it you, Bob, who said on TWiV, if you mix science and politics, you get politics?
That may have been Peter Daszak, but I think that's one of the wise things that he did say. Yeah, you get politics. And, I mean, nobody is naive enough to think that, politics don't play a role in science. And obviously we have to get our funding, through the political process in every country. Yep. But, I mean, science, I mean, maybe I'm just, maybe I'm Pollyannish about this, but I think that we need to have, sort of a dividing line, a firewall between science and politics. And scientists need to be able to collaborate. And they need to be able to collaborate in spite of the fact that, there are political differences between the countries. There's some really excellent scientists in China. I know that because I've trained a few of them. And, and we've been doing that for decades, building that scientific enterprise up there. They spend a lot of money. They're really making excellent things. And, if scientists can't get along and not accuse each other of doing, surreptitious things and hiding data and doing all this kind of thing I think we're in deep trouble, and I hate to see the field going in that direction.
Yeah, that's something we could certainly dwell on for a long time. But back to the data, how did Jesse get these sequences?
Yeah, so there turns out that the deletion at NCBI didn't actually delete where the data was stored. It's the NCBI has recently been set up using the Google Cloud and the Amazon Cloud, so they've recently transitioned to that. And I think what was supposed to happen was that as a deletion request comes in and they execute that then all the data is removed. In this particular case, it turned out that actually the data that lived back on the cloud, specifically on the Google Cloud here, wasn't actually deleted. And Jesse was, because they have a standard file structure, was then able to recapture the data from that structure. Yeah.
Okay. All right. So now we have these. How many sequences are we talking about, roughly? Does anyone remember?
I think it was a total of like maybe 45 or so.
Okay.
Most of them are incomplete. Again, this is nanopore sequencing. It's not for the purpose of doing phylogenetics. It's for diagnostic purposes. I think the final set that Bloom then is looking at, I think, is maybe 10 or 12 or so.
13 maybe.
Okay.
13, yeah.
And these cover spike through the next ORF, right? Is that correct?
Yeah, I think they cover spike and then maybe into 2 off 10. I believe there's some to reach to. Yeah.
All right. I mean, so from the title, he says that they shed light. So what kind of light does his analysis shed?
I mean, I think what needs to be clear here is that getting more data from early full-length genomes from early cases in Wuhan is critically important, and it's something that, I mean, we mentioned as much in our proximal origin paper of last year, where we mentioned that some of the most valuable data we could get is to get genomic information on some of the very early cases, and they haven't been forthcoming. Not because of individual scientists, but I'm sure China again is obfuscating a lot, including some of the early events in Wuhan, which is a problem.
But also those animal markets, right?
Yeah, yes, they're very specifically obfuscating the animal markets. But I think also just getting the samples may no longer exist. The samples might exist, but were degraded. They might not be able to access them for IRB issues. Like there's regulatory issues for why you can't just go back and saying like, look, I really want to go back and get all these samples so I can sequence them. We can all agree that would be incredibly beneficial. And I would say more the really early sequences. So we're talking like December in particular, right? The sequences recovered here seem to be from January and February. So yeah, they are early, but they're not some of the earliest. Yeah. And again, any new data here is going to be valuable. And this particular data adds a little bit to that. I'll say it doesn't really change anything. It doesn't add anything we didn't already know. But more data is always better. Of course. Yeah.
So there's actually a lot of misinformation about that. I mean, even in some of our mainstream media publications, they're saying this data sheds light on cases, the earliest cases of SARS-CoV-2 in Wuhan, and cases before any other case that we knew about, actually, is what the implication was from some of the articles, like in the Washington Post, and some popular Twitter users that like to talk about the origins, have really put out a lot of misinformation about this preprint, saying, look, this proves that the virus didn't originate in the Henan market. Now, Dr. Bloom in his paper has contributed to some of that in a large way, but the reporting has been really, I think, substandard there. I mean, these sequences from January or even February, don't tell us about, it leaked from a lab or anything like that, even though that was the clear insinuation from a lot of the reporting. And unfortunately, I think, that's been sort of shepherded along by people who are advocates of this lab leak hypothesis.
Yeah, and I'll say, maybe just to put a little specifics on it, right, that what we knew based on previous data is that we had two early lineages, which we see in China, including in Wuhan. Mm-hmm. One is lineage A and one is lineage B. And the new data here adds to their lineage A and lineage B. So they add to that existing knowledge, right? Saying that we know that these things are circulating. I think what Bloom does is that he misunderstood a little some of the earlier data. For example, the way that the data is labeled, which is something that for myself and many others who've done phylogenetic analysis, we've already corrected for all these things, that we see already evidence of lineage A and B in Wuhan. Most of them are lineage B. And lineage B is when we see what we detect at the Wuhan market, what was sequenced there is all lineage B. That's not to say that lineage A wasn't at the Wuhan market, because there was probably super spreading at the Wuhan market, right? But we just never captured lineage A because we came in too late. We didn't sequence enough. We didn't sample enough from that market. What we know is from the WHO report, and in fact, we have known this since last year too, where it was reported in some preprints, is that there's this lineage A is associated with a different market in Wuhan. What that market is exactly, we don't know. It's not mentioned in the WHO report, which is unfortunately, I've asked questions about that. Can we please know what this market is? And it's been a blank wall so far, so we don't know. But we already knew that these two lineages were circulating in Wuhan, specifically with the one dominating it at the Wuhan market, right? And then again, the other lineage associated with a different one.
Yeah. So I think that's another problem with some of the reporting, and also, unfortunately, with the Bloom preprint is that, we knew about the multi-market hypothesis from the WHO report. Right. And clearly, the non-market was just part of the whole story, but there were multiple other markets involved. In fact, more than half of the early cases, more than half of the first 170 cases or so, 55% were associated with one market or the other. And they were fairly equally distributed between Henan market and these other markets. So, I think proving that the outbreak didn't start in the Henan market is what the Bloom preprint tries to do. But, it's not even where we're at in terms of, understanding the early days of the outbreak. We know that there were multiple markets involved and even likely multiple spillover events in multiple markets, which would reflect almost exactly what happened in the first SARS outbreak, where there were multiple spillovers and multiple strains of the virus, and probably some diversity developing in some of the animals in these in the wildlife trade before they even got into people.
Bob, he actually mentions your discussion on TWiV. He cites TWiV in the references, which I found amusing.
He popular podcast. In the revised paper, I hate to.
No, it's in the first. Yeah,
it's in the first version, but in the second version, he took it out.
Too bad.
I don't know.
I thought we would get a nice bump, but no.
Unfortunately, he just cut that part out.
But he dismisses your idea that there were two market sources, and then he says the improbable evolution of one of these progenitors towards the other after it jumped to humans. So can you explain?
Yeah, maybe. So the problem here, so it gets really technical. So there's several issues with that particular statement. I'll say the one thing to keep in mind is that the way that these markets, which we now know, right? We know that 4 markets, at least in Wuhan, were trading in live animals, including raccoon dogs that we know from the SARS days is highly susceptible. And actually, we know that from SARS-CoV-2 to highly susceptible for infection and a high probability intermediate host, I would say. Now, these animals are coming from outside Wuhan, and specifically, many of them from prefectures out on the west side of Wuhan, like Enzi and other places where wildlife farming, is highly prevalent. So those animals are coming into the city and then distributed out across markets across the city. It's not just that this market will get those and that market will get all of those. It's that they have a common supply chain, which then funnels into these markets across the city. So you can imagine this situation in which is what we saw with SARS-2, where infected animals come in via the same chain, but then spreading across the city where they're distributed. So when we're talking about multiple spillover events here is, first of all, we've seen it before. Again, we saw it with SARS, and we see this frequently in general, is that we're not talking about like some animals there got infected and some animals there got infected. It's the same animals that come in and then get distributed across. Mm-hmm. So when we are talking about, and I mean, in the Bloom preprint, he talks about a patient 0. What needs to be clear here is that we probably don't have a single index patient, right? We probably have multiple. Yeah. And what we know from another paper by Joel Wertheim and Jonathan Pekka and others is that most of these, like when they come in, probably have multiple spillovers that don't result in any onward transmission whatsoever, right? It sort of gets into people, maybe it infects a person, it infects another person, but then it doesn't get anywhere from there until there's the few lineages that probably by random chance end up being successful. And then those are the ones that then end up starting the pandemic. So there's multiple tries, probably multiple spillovers happening in the early days of this pandemic.
Yeah. So there was another significant paper, and I know that the people that are in favor of the lab leak don't really like this paper so much, but it came out in a Nature Journal Scientific Reports, I think a little bit after we spoke the last time, Vince, and it actually was and maybe we talked about it a little, I don't remember it clearly now, but it's a very significant paper because it talked about, the numbers of animals and the wildlife trade and what Kristian has just been talking about, how they are distributed, throughout multiple markets in the city of Wuhan. Very, very significant paper that, adds a lot to the whole, understanding of how this virus was probably spread to people in this large city of Wuhan.
Yeah, and I think
some of the authors of that paper that came out, I'm not sure I know.
Zhao is the first author.
Okay.
Yeah, and I would say it's very significant because it shows two things. First of all, it shows that live animals that are susceptible to these kinds of viruses were indeed sold at markets across Wuhan, including the Huanan market, but also other markets, which the Chinese government had denied. They specifically said the WHO, and that's what's in the WHO report, is that there were no live animals at the Huanan market. And we now know that that's just not true. So if we're talking about obfuscation, clearly there is obfuscation here, where, again, told no live animals, which were a bit of an enigma for, us looking
really,
into.
It raised important questions like, no animals there, how did it spill over, right?
Yeah. I mean, we knew from prior experience and from data prior to the pandemic. For example, our co-author and colleague Eddie Holmes had visited the Huanan market back in 2014 and taken a photo of a raccoon dog. So we knew that while, in 2014, they were selling raccoon dogs, but Lord knows what that means for 2019. But this paper comes out, complete bombshell in terms of, smoking guns here. This is a big one, right? Because it showed that actually they did sell these live animals and they did so illegally. So that's really important, right? Because it gives you the sort of conduit for like the animals are coming in. None of them were tested. A raccoon dog hasn't been tested for SARS-CoV-2, at least not officially. So none of these animals that we now know were present at these market and high risk of being intermediate animal hosts for this virus. To my knowledge, none of them have actually been officially tested.
But what we do have from the Henan market were the environmental samples. Yeah, and we know that those positive samples were mostly coming from the area of the market where these wild animals were being sold. In fact, it wasn't just, on the walls or, somewhere else. It was actually in the drainage areas where these cages are cleaned out and where, after the animals are killed and slaughtered, where, they rinse those products down into the drain. It was those drains underneath those animal cages that were positive. I mean, okay, we don't have a swab from an animal that's positive, but we are pretty close to having, that very similar thing to that. And with the new paper that we were just talking about, the Zhao paper that, showed the animals. I mean, we're very close to having, the animals being positive in that market.
I want to go back just a little bit to the idea of multiple spillovers and what seems to be, for at least some people or some entities, a focus on finding a patient zero, where I think, we've heard a good case for there being multiple spillovers. But then also, at least in this specific case, how does that relate to the lineage A and the lineage B? I somehow lost that.
Yeah, so this is why it gets somewhat technical. So the whole lineage A and lineage B, when we try to reconstruct what's called a phylogenetic tree, it's basically a family tree of all the SARS-CoV-2s that we have sampled and sequenced, is that we can create a family tree and then we can use phylogenetics to sort of go back in time and understand how they're related. If we're looking at lineage, the split between lineage A and lineage 2 is it's 2 SNPs, it's 2 mutations. And then we know there's a third that sometimes pop up, but it pops up in different parts of the genome. So the split between lineage A and lineage B is 2 mutations. And specifically, one is the C to T, the other one is the T to C mutation. Now, when we're trying to figure out which one of these is the earliest one, right? There are different ways of doing that. Either you say, well, which one did you sample the earliest? And the one we sampled the earliest is from the Wuhan market, and it is lineage B. The other way you can do it is you can say which one is closest to what's called an outgroup, so a distant ancestor of SARS-CoV-2. And here we can use RATG13, for example, which is the closely related bat coronavirus. It's about 96% identical to SARS-CoV-2. We can say, well, if we look at these 2 positions here, what does the bat coronavirus have? And whatever the bat coronavirus have, might assume that that's the ancestral sequence. And when we do that, well, the problem is the difference between Q1, for example, which is one of the first sequences we have and is lineage B, is that there's 1,161 differences between RATG13 and Q1. Now, if we take this presumed ancestor of lineage A, it's 1,159, so 2 less. So the problem is that, and this is where, the issues come in here, is saying that we can't assume that this means because lineage A is slightly closer to the bat coronavirus is in fact the ancestor. Okay. And there's several issues with this. One issue is that we see the virus throughout the pandemic. We see many mutations that actually revert back to what it was in the bat coronaviruses. So something that moved away from the bat coronavirus in the earliest cases, then later mutate back to what the virus was in bat coronaviruses. And many of these, the vast majority of these are actually these C to T mutation, T to C mutations that I talked about. So with this assumption that A must be a root because it is the closest virus is just not true. Because again, we know that these reversions happen all the time. So it could be that B is actually the root or the more proper root of the tree that then evolved into A, probably in an animal reservoir, but could also happen in humans. So when Bloom is talking about this conundrum between the earliest cases being lineage A, but there's others that are lineage A. So the earliest cases being lineage B that we sampled, right? But lineage A is closer to bad coronaviruses. There is no conundrum here. It's just that we are limited in the way that we are sampling, which is just a small fraction of all the stuff that's going on early on in the outbreak. But even the more basic point about A must be the root, so it can't be the Wuhan market, for example, is simply untrue. And it's based on just some misunderstandings of how does the phylogenetic relate to a transmission of viruses out in the human population. And really importantly, how does that relate to probably not just having one spillover, but might having multiple spillovers, most of which are probably unsuccessful.
So unfortunately, the way this gets reported, though, in when it gets dumbed down to its, sort of least common denominator is there. Well, unfortunately, I think, so there are a lot of people that are willing to sort of make that misleading information to the public. Okay, so they're saying, okay, well, this clearly shows it didn't come from the Henan market. Okay, that's leaving aside all the fact that there were all these other cases that were associated with other markets. But, it's reported, okay, it didn't come from the Henan market, and then yada, 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 it must have leaked from the lab. And that's the way it gets reported, right? Okay, so it didn't come from the wildlife market; it had to come from the laboratory. And of course, that is also not true. But I think that talk to some people when you're writing an article for your newspapers. I mean, including the New York Times and Washington Post and some of the other ones, and maybe get the right information and filter it a little bit and get that right information out there. So that clearly did not happen with the reporting around this Bloom preprint.
No, I mean, I think one major issue here, right, is that I'll say like understanding the root of the SARS-CoV-2 tree is very interesting. We have to be careful what we can conclude about what does it mean once we have figured that out. But the problem is, I mean, this is something we have worked on for months because we are very interested.
A lot of smart people, not just you, Kristian, other folks too, right? I mean, yeah.
And it is extremely difficult, right? And very, very technical, especially because we have such a large sampling bias early on in the pandemic, right? Because all the initial cases were associated, that were described associated with the Wuhan market. There was a big focus there initially, probably lost a lot of diversity and other cases that were going on elsewhere. So we just end up not sampling it. So it's a very, scientifically speaking, is an extremely difficult thing to try and do, let alone trying to distill it down into a tidbit saying it came from the lab or it came from the market, right? I'll say this particular study doesn't illustrate either way. It really does not help clarify, which Bloom himself says too, right? That when it comes to understanding sort of what we call the proximal origin of this virus, right? It really does very little to try and resolve that. And again, it is very important that even lineage A, for example, could have circulated at the Wuhan market. We just never captured it.
There was actually a lineage A at a hotel nearby, right?
Right, but yes, so it could be, but again, we have no evidence, right, for it to do, but it could have been. We just failed to capture it. Even if lineage A did not circulate at the Wuhan market, is not inconsistent with the Wuhan market being a major place of spillover, right? Even the only place of spillover, because it could be the lineage B was actually the early virus that then evolved into lineage A. Again, all these things, there's so much uncertainty on the analyses themselves, but that adds to additional uncertainty when we're trying to say, so what does it actually mean?
Yeah, right? Yeah. But Kathy, to get to your point about what does it lineage A and B imply for the lab leak hypothesis or for the natural origin hypothesis. I mean, both of those, now competing theories or hypotheses or whatever. I hate to call the lab leak a hypothesis even because it's basically unfalsifiable. You can't prove it one way or the other. But okay, let's give it a hypothesis designation. if you believe that it did leak from the Wuhan Institute of Virology or some other lab there in Wuhan, you still have to account for the fact that there are these two different lineages, right? Now, the natural spillover, multiple markets, or multiple spillovers of multiple markets, that's a pretty simplified, straightforward way to get these different lineages into different different markets, different populations. But if you believe that it's a spillover from the lab, then you have to go through, okay, it came out of a sample, went through somebody working in the lab, and then that person somehow or the other ended up in not just one market with one lineage, but into another market with another lineage. And you really have to cobble together a lot of, very unlikely events, highly unlikely events to end up getting, most of the early cases, 55% or even more of those early cases associated with the wildlife trade. So, if anything, the Bloom preprint adds to the, growing data, and there's actual data here. We're talking about epidemiology and genetics and everything for the natural spillover hypothesis. If anything, it points you even further away from the lab leak hypothesis.
So one of the papers cited, and we talked about before, is this Kumar et al. From Molecular Biology and Evolution, where they did a different kind of clock calculation, and they come up with pro-CoV-2 circulating late October, early November, right? I was wondering if you could give us your thoughts, because Bloom says some of his sequences are close to that one, or identical at least.
WA1. Yeah, go, Kristian. Yeah.
Yeah. So pro-CoV-2 it's essentially to do the version I said, where you say, well, the one that's the closest to the bat is the most likely ancestor, which I agree is the most likely ancestor, but because of these reversions and sampling biases, it might not be.
Yeah.
Right.
Okay.
But in that Kumar paper, they're saying like, okay, look, we have these three positions where this version of it is closest to the bat ancestor. So we're going to call that the ancestor. Mm-hmm. The thing is, the pro-CoV-2 is actually just WA1, which is the sequence that was sequenced in the United States, from Washington State specifically, via recent travel to Wuhan. That has the same three SNPs that we are talking about here as pro-CoV-2, and is from Wuhan. So again, whether you say pro-CoV-2 or whether you say W81, I'll say that I have previously speculated back when I was on Twitter that I thought WA1 was probably the best route for the tree if we had to pick one. I've sort of come a little bit away from that because, again, all the stuff we have learned about the reversion, so I'm not actually so sure. But that's what that is. And the problem is the way that they get to pro-CoV-1 and then saying like, oh, well, if we fix that as the root and then we're going to do the tree based on that, it ends up being a circular argument because the tree you're trying to resolve is the data you use to create pro-CoV-1 in pro-CoV-2 in the first place, right? So again, it's really technical, but none of these really help us try and resolve what is a proper root of this particular phylogenetic tree for SARS-CoV-2.
Yeah, but it does get to the origins question. And I think this is where the WHO report was especially valuable. I mean, the letter to Science from how many ever it was, 16 or 18, I don't know how many we have left now that actually are supporting that letter, but for Science said that the WHO report didn't add anything to the understanding of the origins of SARS-CoV-2. And that's definitely not true. I mean, there's a lot of data in there, including a lot of data we've talked about in the past hour here now about the origins that, it's extremely valuable in learning. And we shouldn't, dismiss that just because, there were some Chinese politicians in the room when some of the discussions were going on. I mean, that data, you have to be a scientist. You have to look at the data. You have to evaluate it and, see if it makes sense. A lot of it makes a lot of sense, and it adds a lot to the understanding of how this virus spilled over.
So let me ask you, for SARS-1, we got to a 99% identical virus in a collection of sequences in a cave that could have constituted SARS-1. So can we get to that for SARS-CoV-2, and how do we do that?
Yeah. Yeah. Actually, Vince, I think the closest we got was about 96 or 97. Am I wrong about that, Kristian? We never got to the 99% virus.
We got the ones in civets were pretty close.
The ones in civets, of course. Those are the intermediates. Yeah, those were a couple snips away. But,
well, more than that. But again, I'll say, what is probably, based on just all the available evidence. And I'll say that the Bloom preprint, I don't really think adds to like our understanding of the origin, right? I'll say the major new understandings we have with like the epidemiological data and the animal data and things like that. Now, when we're talking about SARS-1, is that did spill over multiple different times into the human population and was clearly widespread in the intermediate host population. The animal traders had sky high seropositivity rates, the animal tested positive, like all these things, is that SARS-1 was clearly widely distributed in an intermediate reservoir host. The same we haven't seen for SARS-CoV-2, right? And there's a couple of different likely reasons here. One is that during the SARS-1 epidemic, they actually did not close down the markets immediately, right? They allowed the wildlife trade and everything to continue, so you could have additional exposures that way. That got shut down for SARS-CoV-2 pretty early on, right, by the Chinese authorities, both the wildlife farming itself back to the supply chain, but also the markets themselves. So that might have limited additional spillovers that could have been helpful in terms of identifying potential intermediate hosts. Another issue is that if you're just looking at reported data, they haven't tested these potential intermediate hosts, right? Because they denied that they were even there in the first place. And this is where I'm really concerned around just where the origin investigations are going, because it's become so politicized and so focused on a very specific hypothesis that explains part of the emergence of SARS-CoV-2, specifically the lab leak hypothesis, that we are forgetting that there are the other theories that are more likely, right, just from the get-go, and actually they're supported by available evidence, is that we are failing currently to investigate those properly and to put pressure on China and saying, look, why did you say the animals were not at the Wuhan market, where in fact now we know they were? Where exactly did these animals come from? Did you test those farms? Did you test these animals? Did you test them at the markets? Did you test them back at the farms? Have you done serology in the wildlife traders? Have you done serology in the people working at these farms, for example? All of these are things that need to be done so we can try and figure out, is there an intermediate host that we can actually identify there? Right now, it might be being done, I don't know, but none of it is being reported on, right? And China is being very quiet on this, saying like, nope, we're not, this happens outside China, which is a big, big problem, right? Because this is a very likely conduit of how the virus came in and then funneled through into the markets in Wuhan.
And it will happen again. I mean, multiple spillovers happen all the time. I mean, MERS coronavirus, which is a virus of camels. It's not a human virus, right? It is a camel virus. Our friend Andrew Rambo, makes that point a lot. It's a camel virus, not a human virus. But those spillovers are so numerous that, we've lost count of those. And the reason why, MERS has not become a pandemic virus is because you can't maintain those transmission chains. So basically, most of the new cases of MERS in humans are, new spillovers, directly from the camel reservoir. So
I will say, though, just in terms of intermediate hosts, we have learned a lot of just, the other viruses that look a lot like SARS-CoV-2, which we have learned during the pandemic, found in bats and pangolins, and it's quite remarkable how much diversity there clearly are in these subecoviruses, right? And I will say that more than anything is terrifying to me when you're thinking about the wildlife trading and the wildlife farming. I mean, pangolins are being bred, right? Not very successfully, but they are being bred, and certainly things like raccoon dogs for both for human consumption, but also the fur trade. All of this is happening, and all of these animals already carry viruses that are not SARS-CoV-2, but they look a lot like SARS-CoV-2. And the question is, what are the ones we are failing to detect right now? What are the ones that we haven't actually seen yet? That might be the SARS-CoV-2 waiting to hop over, right? And that's why it's really so important that we go back and we're saying, like, look, let's just start at these animal markets in Wuhan and trace back in time and saying, what do we think could have happened here? Again, where did the animals come from? What animals? Were they tested? All these things, right? All of this is really something that needs a very significant focus and needs to being done, because that's the real risk for continuous spillovers. Again, the SARS-CoV-3s and the SARS-CoV-2s, right? Yeah. Is that we obviously need to understand that.
Let me follow.
But Kristian, some of those things you're just saying there, some people are saying, I'm not going to name names here because to give, but you'll find them on Twitter talking about how we need to shut this type of research down, to actually not go and look, to see what the diversity of the coronaviruses is in nature or in bats or in other species. They say that's too dangerous. And, we should just, sort of put blinders on and hope that, they don't spill over again, I guess. I guess that's the alternative. It's really quite the opposite of what we need to do. And, regulate labs. These labs are already very highly regulated. Maybe there needs to be some more international agreements, but the route we're going is not going to give us those international agreements. That's for sure. To talk about what needs to go on. But these labs are secure. They're widely built. We just had a scientist from Australia who had worked in the Wuhan Institute of Virology right up until November. A very brave person coming forward, because she's already been viciously attacked by, people who believe in the lab leak hypothesis that, say she's, just in the arms of the Chinese Communist Party. What does she know? She's lying to us about what went on there, and she basically just came out and said, look, it's a very well-run facility. They weren't doing gain of function research there that she was aware of, and she was interacting with the people in the institute. There was nobody sick there in November that more than just your normal flu or anything like that. There wasn't widespread SARS, COVID-19 there that she was aware of. And, these are the types of things that, I hope the intelligence community is actually going to talk to her and listen to what she said about, what was going on at the Wuhan Institute of Virology in late in 2019. That's probably, her words are probably more reliable than, looking at, say, the numbers of cars parked at the hospitals in Wuhan and things like that have been pointed to as, evidence that, there's a big cover-up going on.
Kathy, go ahead.
Kristian, you mentioned something that I heard Peter Daszak talk about in another forum, and so you've really re-emphasized that, and that's that China closed out these wildlife farms right away as well, and that seems like a key thing to try and get at understanding this. And I had a question here in the notes about, if in an ideal world, if there were no politics and you had all the money possible, what would be the most important steps that you would want to take? And I guess you've already touched on them, but I just found that wildlife farming link to be intriguing.
Yeah, I mean, I'll say from my perspective is, right, that we need to do everything, right? And we need to do so collaboratively. The problem is that we are on a collision course right now where we're not going to get any cooperation. And the problem with that is that we need that cooperation. The sense from, this is in the Bloom preprint too, that there's all this amazing gold mine of data waiting on deleted servers so we can figure it out without the help of China. Unfortunately, that's not really how this is likely going to work, right? And my concern is, again, that drawing up accusations, mixing up obfuscation with covering up and understanding why obfuscation is happening makes it very, very difficult to work in an environment with saying, look, we need to work with Chinese scientists. We need to work with the Chinese government, but that's the politicians' work. That's not our work. to try and figure out, again, trace back in time of like, let's go back to the really early cases. Let's understand the very early cases the best we possibly can, and then trace that back in time, right? And again, of course, keep an open mind, right? As you have all seen my email to Tony Fauci, and I myself have considered very soon, so has Bob, very considered the possibility of a lab leak. But here's the thing, is that a priori, it's an unlikely hypothesis. But when you then update those priors based on available evidence, it's clear that it comes up empty every time, right? There is actually no evidence that could have to do with a lab leak. The evidence we do see come up has to do with obfuscation around the animal trade, the animals actually being present at markets, early cases. And it's not just early cases that cluster around the Wuhan market, I should say. It's actually also excess deaths. And again, the virus is actually there, right? All of which is clearly pointing strong epidemiological links. It doesn't mean that we have now disproved the lab leak hypothesis. It just means that something that was unlikely in the very beginning is based on current evidence is now even less likely. But again, we need to keep an open mind, right? But we need to focus and continue to study all hypotheses, but with a focus on the ones that are by far the most likely, and which additional evidence, like where are these animals coming from? Did you test at those farms? Did you test the people working there? All these kinds of things, right, is really what I would like to see a renewed focus on. And again, I don't think right now, I think it's totally being neglected. Nobody is asking questions about animals, right? People are asking questions about protocols and gain of function research, for which we, as far as I know, there's no evidence of published gain of function research at the Wuhan Institute of Virology, right? So it's all of this is mixed in that just makes it really, really difficult to honestly get studies done that would put us further towards understanding how did SARS-CoV-2 emerge in the human population, right?
I also wonder, because your history is in looking at outbreaks and early spread, how much different this is because of asymptomatic infections. Has that been a factor in the other viruses? And just, I'd like to hear you talk a little about that.
Yeah, I'll say, I mean, it's certainly, I think it sort of complicates some of the interpretations. For example, let's go back to the early cases in Wuhan, right, where most of them are associated with the market, but not all of them. And I think a lot of people have taken that to terms like, well, unless all of them came from the market, it can't have come from the market, right? And that's just not true for the reasons that you just mentioned here, that while the association is clear, but it's not perfect because there's also transmissions out in the community. And by the time you detect it, for example, detected it with cases that then realized was associated with a market. So it's not that there was increased surveillance at the market, right? It's just the association came later. Is that we would never expect that to be a perfect association between all the early cases came from the market. And SARS-CoV-2, unlike SARS, where the same thing was true for SARS, I should say, not everybody was associated with markets either, the human food chain, right? Is that that's even harder for SARS-CoV-2 because it can spread asymptomatically and because it can spread before people get symptoms. So again, these things that are drawn up as being mysteries, and even to say like it couldn't have come from a market, just isn't true based on what we know about this particular virus. And it certainly makes it more complicated than many other viruses where we were trying to do the same kind of investigations.
Yeah. In terms of the epidemiology, I mean, if it had leaked from the lab somehow, I mean, you would expect to have seen cases, near the lab, which is about 10 miles, 20 kilometers or so away from the center, the central business district of Wuhan. It's it's not in the center of the city proper, but a little further out in the provinces. No epidemiological link at all to anyone in the market. You had those famous, 3 cases from the Wall Street Journal, which, people like to focus on. We don't even know that those existed. They were in November during a surge of influenza. Who knows if they exist or not? Did they have COVID-19? Unlikely, because if they had and they were sick enough, they would have probably been another 100 or so cases at the institute. We know from the other Dr. Andersen, Danielle Andersen, she's got that O instead of an E there, Kristian, but it's okay. And, we know that there wasn't a huge outbreak of respiratory illness at the Wuhan Institute of Virology in November of 2019.
Yeah, and I'll say, that type of data is what's important, right? If we just look at Wuhan, the Huanan market is north of the Yangtze River, and the Wuhan Institute of Virology is south of that, about 20 kilometers away, right? And again, if we just look at where do we find the early cases, well, again, as I mentioned, they all cluster around the Huanan market based on retrospective contact tracing. But of course, there is a bias there because there was a lot of focus on that particular market in terms of case identification early on, right? But that's why things like saying, yes, okay, so it's not perfect data, but then that's why when we say, well, look at excess deaths, which then happens later on, that goes into January, right? Is that it's the same picture where all the early excess deaths here cluster north of the Yangtze River, specifically where you find the Huanan market, and not south of the river where the Wuhan Institute of Virology is. So it's not that the virus is moving away from the Wuhan Institute of Virology and then across the city. It's actually moving towards it from the Huanan market. But that's why it's so important. And I will say, these 3 illnesses, I don't know what to think about that, but I need to know more, right? Because if there were 3 illnesses at the Wuhan Institute of Virology that were consistent with COVID-19, that's important knowledge. But the problem is, again, that we don't know because the intel seems flaky. It's been many versions of this have been out. There's reasons why people go to the hospitals. That's where the doctors are. It's in the middle of flu season, like all these things, right? But that's the kind of knowledge which is really critical, both as it relates to the Huanan market and other markets, but across Wuhan in general. And I would say really importantly too, outside Wuhan. What about these prefectures and small cities that they were doing wildlife farming, where we know that they were sourcing animals into these markets in Wuhan? Have we gone back there and looked for evidence of infections, whether that with serology or case study, case definitions and things like that? And that's why it's just so important that we just get as much additional data as we can get so we can get clarity on the most likely explanation for what happened here. As the data stands right now, it's highly suggestive, right? You look at the emergence of this in November, probably, of 2019, SARS emerged in November of 2002 in Guangdong, Foshan City, right, which is about 1,000 kilometers away from Yan'an. Wuhan is about a little more than that, right, but far away from Yan'an too. But the similarities, the timing, big Chinese cities, wildlife markets, the timing around the winter months here, it's all the same. And HKU1, I should say, is the same thing. The first case of that was detected in, I think, December or January of 2004, 2005. Unknown pneumonia, so it's the same picture.
With a pure cleavage side
going on over and over again.
So, and HKU1 is influenza, right?
No, that's a beta coronavirus. So that's another human. That's one of the common cold coronaviruses. Yeah. And I should say that it doesn't. That was the first detection. That virus is clearly older, so it's been in the human population for longer than that, right? But the similarities were, again, novel coronavirus at the time found in humans causing pneumonias. And then you sort of trace back in time and see that the similarities here are really striking. Yeah. But I will say, because a lot of people have this like, well, but you're saying that you think the emergence from animals is the most likely because that's what you've seen before. And it's true that that's why we base our priors on, but we have additional evidence, right? Again, the early cases clustering, the excess deaths, the presence of the virus in the environmental samples, the multiple lineages in multiple markets, like all of these things, which actually adds additional evidence. It doesn't prove it, right? Because it's very, very difficult, if possible at all, to prove anything here, right? But that doesn't mean that both of them are equally likely, and we should continue to suggest them, to be equally likely until we have full proof of one or the other, right? That's just not really how it works, in my opinion.
So with respect to these 3 putative cases at the Institute, we asked Marion Koopmans when she was on TWiV just before you were, Bob, and we asked her and Dashak and Fisher, and they said no, they were not COVID, they were something else, but they said no, definitely not.
And Danielle Andersen, added a lot. Even she's being attacked a lot, but she was there. Dr. Shi was in November going to a conference in Singapore on Nipah virus. I mean, she wasn't concerned about a huge lab leak that was spreading throughout all the staff at the WIV. She was there at a conference, and it just those 3 cases are a red herring. I mean, there's a lot of that bad reporting out there. I mean, I think you're going to see. I mean, right before our TWiV here, there was apparently some sort of a side conference in Congress about, CGG codons and things like that have gotten a lot of attention recently. And that is just really bad information. I mean, if people are going to report on that, they need to actually talk to experts that, can tell you that, that's not such an unusual thing at all, that in fact, furin cleavage sites are present in a lot of beta coronaviruses. Yes, a lot of beta coronaviruses have furin cleavage sites, and it's 4 of the 5 subgenuses or subgenera of beta coronaviruses have members that have furin cleavage sites. It would be unusual if you didn't find sarbecovirus that had one eventually. So just get the facts and report them, and It'll all work out. The science will drive this eventually. Right now, it's just a narrative, a false narrative, I would say, that's driving a lot of discussion.
So I was at that meeting in December in 2019, the Nipah conference with Daniel Andersen.
Oh, really?
Yeah, I was
wow.
there. That's where I interviewed Dashak for the first time.
You didn't catch COVID-19 there, did you?
I did not.
No, and I should say, just one thing to make from my perspective, right, that I think, again, these 3 cases, which are being talked about a lot, and I don't have any grounds to believe the data. I don't have any grounds to fully dismiss the data. I see a lot of inconsistencies that make me very suspicious of that particular data point, right? But it also has to be clear that if you actually, let's just say that you actually had 3 cases of COVID-19 that required hospitalization at a single lab in November, at the Wuhan Virology, you would have had a huge number of infected people at that time to have 3 people that will actually end up in the hospital, right? So there's a lot of inconsistencies here where you're sort of like, it doesn't really make sense. But I maintain still, though, that I want to see what is that actual data. I don't want to read the Wall Street Journal article about it, right? Which is recycled every 3 months. I want to see the actual data. So where did you get that from? Exactly who were these people? What symptoms did they have? Why did they end up in the hospital? What is the explanation for this? I do think that these things are important, right? And needs to be, but it has to be in a sort of atmosphere like collaboration, right? It's like, look, we're just trying to figure this out. If it's the accusations, I mean, nobody's going to volunteer that kind of information, right?
So I think the 3 cases originated in this bogus memo from Mike Pompeo that he released when the administrations turned over, right? And he said, he wanted to have a last dig, and he mentioned this, but there's no evidence for them at all. I think it came from.
No, I think my understanding is that it was actually first reported by an Australian newspaper, and then yes, it made its way into into that memo. Yeah. And I'll say, I mean, that memo also says things like the Wuhan Institute of Virology were working on pangolins. And I don't know, where does that information come from? Because working on pangolins at a lab, that's pretty difficult. I mean, it's not impossible, right? I mean, anything is possible, but it's like a bottomless pit of hypotheticals here. Is that what's the data showing that the Wuhan Institute of Virology were working specifically on pangolins? Because if they were, I want to know that too, right? I want to know everything that we can know, because it's important, right? But I think just a priori, it seems unlikely, right? Why would they work on pangolins? What did they do exactly on pangolins, right?
Why pangolins? I mean, they're cute, but I mean, they must be hard to work with in the lab, right?
Yeah, it doesn't make sense, right? And again, that's why it's important that, we just need to understand that what are the sources for this level of data, because it does not seem that you can actually read too much into any of that.
Yeah. Well, the pangolin looks interesting in hindsight, right? Because now we know they harbor similar virus, but they didn't know this, so why would they work on it? So in light of everything we've said, I mean, it seems like this is not a paper that should be published, but I'm sure it will be, and it'll be met with another round of unfortunate press, right?
I mean, I think, again, there is a version of this that I absolutely do think should be published, which is that, you say, look, guys, I found this data and it's actually quite helpful for my analyses. It doesn't tell us anything about the origins, but it's a data point. So let's put it out there. Let's work on it together and then, get it out in the published realm. What I really object to is this accusation of misconduct, right? I mean, how anybody can come to the conclusion that there is no plausible scientific reason for the deletion, it therefore seems likely the sequences were deleted to obscure their existence. That's the part that was like, first of all, that's not science, right? There's no evidence to suggest that they did this to obscure the existence, right? Talking about this deletion as a failure of science, again, these are the things that is unnecessary. They're non-scientific, right? They don't help drive what does this data tell us and what does it not tell us, right? So I would certainly like to see a version of this, which is done in collaboration and saying like, look, we developed this nanopore assay. We didn't realize the sequences were a value at that time, and we had concerns about quality or whatever. But now we resurfaced it because we think maybe, as Jesse points out here, maybe it could help us resolve some of the rooting of the SARS-CoV-2 tree. I mean, absolutely. More data is always better. Yeah. Right? But that should be the focus.
Yeah, and please, I mean, just to plead, don't thank the so-called internet sleuths and other dilettantes and people that have come out with the lab leak hypothesis that want to write books and other things about how bad the Chinese scientists are. Stick to the science, please. I mean, really, there's been very little that has come from this internet sleuthing. Let's the cave, the Mojang cave, and other data. The viruses in that cave are not close enough to SARS-CoV-2 to have been the progenitors. Those kind of things. So just a lot of distractions there. Why do we need to thank those people for spinning up all this stuff?
Yeah, that's a good point in the acknowledgement. He does thank Twitter discussions, which seems.
And I will say, I mean, I'm all for sleuthing, right? I think what has to be clear though is that what can we conclude based on the kinds of evidence that we might uncover, where we have no insights to motives, we have no insights to why they did one thing and not another, right? And I'll say, unearthing of some of these older theses from TWiV. I mean, there's one from 2019 where they sequenced a bunch of bat coronaviruses, and guess what? None of them were SARS-CoV-2. I mean, it's one of those things that are saying like, sure, I mean, it's all good, right? Let's look at everything that's out there, but we can't then take that and push it into a narrative that they're all covering up, they're all lying. So when they're saying this, they don't actually mean that. They mean this stuff over here. They didn't do things perfectly. So that's a big red sign there, right? That's the part that I think we have to be really careful that we don't get down that line of inquiry, but instead focus on what's the actual evidence, what's the actual data, what are the conclusions that we can and cannot make based on available evidence.
Yeah, don't thank the people that are attacking the scientists, like Kristian, who I think probably still has, a lot of really bad people coming after him, and other things like that. Don't thank those people. I mean, that's not science. That doesn't belong in a scientific paper.
All right.
I was just going to say that the title of the paper, "Recovery of Deleted Deep Sequencing Data Sheds More Light," and I actually think that it sheds more heat than light.
Yeah, that's exactly right. Yeah.
Well,
we talked about forests and trees. I think this paper is basically a TWiV.
I think you've mentioned a lot of the inappropriate language, like the first sentence, shrouded in mystery. I mean, that's not something you would write in a science paper. Anyway, I think this was a good discussion. I'm sorry to have gone over, but you guys were into it, and you didn't ask to leave, so that's great.
We've been into it for about the past 16 or 17 months, haven't we, Kristian?
Yeah, we've been digging at this question for a while.
All right. Well, I want to thank you both. Bob, Garry from Tulane University, thanks so much for coming back again. You're probably going to be here again, so.
My pleasure, Vincent. Thank you so much. Yeah, this is not over yet.
Yeah, and Kristian Andersen from Scripps Research, thank you so much. Good to have you.
Yeah. Thank you, Vincent. Thank you, Kathy.
Yeah. And, thank you, Kathy, too. Great questions. Thank you.
Look forward to having you back. We'll talk about West Nile sometime, Kristian, okay?
Sounds good.
All right. Bye-bye, guys.
Thank you, everybody.
One of the things I learned, hadn't heard of, was the excess deaths and where they were in Wuhan. I found that interesting. So, new information.
You never know where a conversation is going.
Right.
I've learned after 12 years of TWiV that make no assumptions.
Right.
It usually is going to turn out better than you think. I thought that was very useful. And I mean, the thing is, it's an hour and a half of talking about it. And if they talk to a journalist, 10 seconds, one sentence or two would end up, and the rest of the story would be spun by the writer. And that's just not how you get the whole story. Right. But I have they answered a lot of the questions before I even asked them. A lot of the things that I wanted to talk about was really good.
It was.
And Andersen left Twitter. I don't blame him. It's a cesspool. I come and go. All right, let's do some picks and wrap this up. What do you have for us?
I picked something. It's an astronomy picture of the day, and it's a picture of a Steve over Copper Harbor, Michigan. And Mona, who's a TWiV listener, sent this to me a while back. And a Steve is a strong thermal emission velocity enhancement. And they've probably been seen since antiquity, but it's only recently been realized that their colors and shapes make them different from auroras. So they're sort of pinks and purples. You think of auroras as being those greens and yellows. Anyway, so Steve's, and then this is really interesting one over Copper Harbor. And then when you click in the caption on Copper Harbor, it takes you to this beautiful video of Copper Harbor, which is the northernmost part in Michigan. Beautiful. So the very northernmost part of the Upper Peninsula. And so one of those nested pics that Rich often have. Yeah,
it's gorgeous. Yeah, very nice. My pick is an opinion piece that Amy Rosenfeld and I wrote, was just published in the New York Times. It was published online Sunday, the 27th of June, and then the paper copy came out on the 29th.
28th.
Yesterday. Today's Tuesday, yeah. So I just wanted to talk about this for two reasons. First, the current title online is "Human Behavior During the Pandemic is More Important Than Any COVID Variant." I may tell you, we went through multiple titles. This is not the one we wanted by far. Amy had a great title. She wanted variants, scariants, and human behavior of concern. Of course, they nixed that. They went through a whole bunch, and here's what's interesting. They said they take a few titles and run them by their social media team to see which one works best, which I find completely, I don't know what the word is I should use.
Yeah.
They run it by some people who say, yeah, that's a good one, or that's not a good one, instead of what the authors want, right?
Right.
Plus, the whole article, it took weeks to write this, and it went back and forth, and they kept changing. They took all the science out because they told us it has to be understandable by a fifth grader. So most of the science is out, which really waters it down, unfortunately. You're going to get the bottom line, which is we think that, just get vaccinated. The vaccines will work against variants, and we go through some analogy with flu and so forth. It's under a paywall, so I'm sorry if you can't get it, but send me an email and I'll send it to you.
Yeah. Also, talking about headlines, then in the paper copy, the headline is just vaccines also fight variants. So it doesn't talk about human behavior at all.
Yeah. That's not the point of the article. Actually, the point of the article is that the outbreaks are driven by human behavior, not by particular properties of the variants, which takes years to study and confirm and so forth. And so on Friday at about 5 p.m., I got an email. So we had been emailing with an editor here in New York she was very good. She understood our points and made good changes. And we went back and forth in the previous days. And then we got an email from another person who was dealing with the print version. And she said, this is going to come out in print on Monday, but it has to be shortened. There's no arguing. It has to be shortened. It's like, so.
Yeah, it's one column tall. So that was
cool.
That was the page.
It's pretty long still. But she took out a lot of things, and she wanted to take out the information on influenza. She said, everybody knows that. I said, no. Everyone does not know that flu variants are more fit and not more transmissible. We need to leave that in. So we took other things out. But in the end, I'm afraid it gets watered down a bit.
It's a good op-ed, and it sees a lot of people this way. So, yeah,
I think people are sort of getting the point. The lady we were working with said it's doing well, so that's fine. And we wanted to get the message out there. At a time where someone told me yesterday, someone said if you pass someone in a hallway and they have the Delta variant, they can infect you, whereas before the other variants would not, which is just totally bogus and absurd, right? I don't know where this comes from.
Yeah,
but
pseudoscience. It's frustrating.
one more thing I wanted to tell you. So at the bottom of the online, they have a little more information about us because they can't do that in the print, right?
Right.
So they said you can have your affiliation at the top, but then if you want, you can add stuff at the bottom. So online, they said, Amy is a virology researcher for 2 decades, and I wrote a textbook and do TWiV, and I'm amazed they left TWiV in because, we had originally written, Amy is a lab rat who spends most of her time researching viruses. They took that out immediately. They didn't want that. It's not serious enough for the New York Times, right? So I have written in letters before, but I'd never written an opinion. So it's an interesting process. And you kind of understand what journalism is up against, right? Especially with science, because you have people who want to, it reads like a Times opinion. It doesn't read like how we write at all, I think. I'm not sure that there's any personality coming through because they want to change it to fit their style and what they think is their readership. And I'm very sad that they think it has to hit a 5th grade level of science education. Yeah. Isn't that really not good?
Well, and it also drives home the point, which we have heard and talked about a number of times before, is that the authors don't write the headlines.
That's right. It's not good because the headlines are what get you, right?
Right. And that for now we know that for online, it's not even editors that write it. It's social media marketers that focus groups decide your headline.
I wonder, what do they do? They have like 50 people they send different headlines to, and they vote on this one or that. My gosh, it's not good. Not good. Oh, well. But check it out. As I said, if you can't get it, let me know. I'll give it to you. You got a nice paper copy there. Can you hold that up again? Let me see what that looks like.
I didn't trim it, but Yeah.
Vaccines also fight variants. Cool. It takes the whole side of the page up. That's cool.
Yeah, yeah. So I wrote all the extra stuff here, the other title, and then the extra stuff about you and Amy at the
That's
bottom.
interesting.
I wrote in.
So it's funny, when we go to Ian's lab across the street, he's written a bunch of those, and he has them all framed on the walls of his lab, right? The paper copy framed and stuff like that.
Yeah.
It's cool. All right. That is TWiV 774. You can find the show notes at microbe.tv/TWiV. You can send your questions and comments to twiv@microbe.tv. And if you like what we do, giving you good science information, mainly viruses, you can think about supporting us, microbe.tv/contribute. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.